Okay, so now that I've explained how the primary productivity lab was set up, I'll go through the data and questions. So there's a lot of please read through this introduction. It really covers a lot of the science behind the ecology behind looking at doing a primary productivity lab. Of course, here's a Khan Academy picture I just included. Again, just showing the idea that the sun, energy from the sun is getting converted to chemical energy by plants, and that every step along the way, heat is being lost to the environment as energy moves from one trophic level to the next, to the herbivores and the carnivores, the primary and secondary consumers, and then as those things die and they decompose and so on. So it just kind of goes through again the photosynthesis, respiration equations. Again, we're looking at oxygen, and of course oxygen is a byproduct of photosynthesis, but it's also a reactant of respiration. Okay, and then again, just the idea of the trophic pyramid, right? So representing the fact that you just have a lot more primary producers than you have primary consumers, and so on. As you go up the point, is it going to be an apex predator because there's just such a small amount of available energy left at that point. All right, so the procedure talks about that. So here's the data table that you can go ahead and screenshot this if you want to from my my video here, or just pause and copy the data down. I'm just going to go ahead and say these are the values that we got for one of the classes, and of course then these are the conditions, right? So container number 1 was just getting the initial dissolved oxygen values from the pond before we put the water into capped containers, and then we had 100%, 50%, and 0% light depending on the condition, and then the dissolved oxygen values. So just looking at the raw data, of course, let me get a different writing tool here. I'll use maybe this one. Just looking at the raw data, so in the light, it looks like we had net photosynthesis occurring because the oxygen levels increased. Looking at the dark and the partially dark environments, looks like we had a net loss of oxygen, because even though maybe photosynthesis was occurring here, just not very much, and more cell respiration was occurring in here, we only had cell respiration occurring. So that's kind of the way I look at those values, just the raw data. And it tells you how to calculate gross net productivity. So for instance, for gross productivity, I'll just choose this one here. For gross productivity, I would take the DO of the light experimental tube minus the DO of the dark experimental tube divided by 24, because I just ran this overnight. So 24 hours, we'll just say. So if I do those numbers and crunch them, then I get a gross productivity of 0.016. So I'm just going to write all my answers as 3 decimal places to compare them. It's just a lot easier to compare them that way. Net productivity is, again, it's a little different calculation. It's taking the DO of the light experimental tube, that would be this one, minus initial DO, and then dividing that by 24 hours. And so then if I do that, then I would get that value, for instance. And it's negative. Again, it's negative because in that tube, the net productivity of oxygen decreased. So we had more, again, oxygen left after respiration. There was less oxygen left after respiration because the number is negative. So to calculate the rest of those. This experiment was the dependent variable. What we were measuring, right? We were measuring, so that's maybe tricky. We were measuring these 3 columns there. But I would say that the dependent variable is productivity, right? So whether it's gross productivity, net productivity, it's the amount of photosynthesis that's occurring, and we're measuring that as productivity. You could also say, I suppose, that the dependent variable is the raw value, the dissolved oxygen value that you're recording. Independent variable, of course, is the setup, right? How much light was being received. Oops, I never ran a negative control. What would that look like? Well, a negative control is going to look the same, except you're gonna have like the same containers, the same amount of water in the containers, but we want to run a negative control so that the values are all going to be the same. So we'd want to think about how can we run an experiment where really, why are those values different? They're different because of the reaction of photosynthesis and cell respiration. So how can we run this experiment without those being, well, we could run pure water would be one example. Or we could like with our enzyme controls, so when you're rate of enzyme reactions, we would maybe think about, well, let's heat or denature an enzyme. So we could do that. We could kind of like boil the pond water, maybe to kill off the organisms so that they're not able to do photosynthesis or cell respiration. But we would still have to run the 3 different situations, 100%, 50%, and 0% light, but the water inside the containers would not contain organisms that were able to do photosynthesis or cell respiration. Calculate the respiration rate. It's already kind of done. Respiration rate is really the dark tube, because that's all it's doing is cell respiration. So, did photosynthetic activity add oxygen to the water? Did photosynthetic activity add oxygen to the water? Yes, because 10.51 is greater than 9.95. Again, that was in the full light. Did aerobic respiration occur? Aerobic, what does that word aerobic mean? That means that O2 is being used in the mitochondria in the electron transport system. So to help generate ATP, to be part of that process. Okay, so did aerobic respiration occur? Well, if it did, then we would see oxygen levels decreasing. Okay, if aerobic respiration occurred, because oxygen is being used for that process. So looks like it is, right? Because that number's lower than the initial. So yeah, aerobic respiration occurs in pond water. What effect did light intensity have on primary productivity? The effect is that light intensity increased primary productivity. Full light gave you a higher DO value than some light. And number 8, so number 8 is like how all these factors might affect primary productivity of a pond. So I just thought it might be better to see some pictures. So I'm not sure if these are in order or not, but one of them was like, maybe this is like the last one. What are zebra mussels going to have an effect on? Well, zebra mussels are an invasive species where I live. And so, they operate by siphoning out those plankton and primary producers, right? So algae and bacteria and stuff like that. And so they're going to clean the water, which, that's nice. They're going to have nice clean water, but it changes the whole food chain. And so it's going to decrease primary productivity because they're so efficient and they have no natural predators. And so they really change the biology of the lakes and bodies of water that they're in. If you have a bunch of like fertilizer runoff, well, you have an initial algae bloom, so that'll increase primary productivity initially. But then, unfortunately, you have this dying off of the algae, and as things decay and go through decomposition, then dissolved oxygen levels will be very, very low because cell respiration is occurring. Pollution into the stratosphere or for like muddy water, that kind of thing is one of them too, is going to decrease the amount of available light getting through to the producers. And water temperature also has an effect. So cold water will have a lower primary productivity, and then you raise the water temperature, you increase the productivity, but at some point it gets too hot and things don't do as well. And so there's kind of like this perfect or ideal temperature it depends on the organism, right? So it's going to change based on the organism. All right, and then the FRQ.